All right, well, welcome back. This is part 2 of our video, our lecture on RNA and protein synthesis. So in part 2 here, we're going to go over protein synthesis now. So what is it? Well, synthesis means to put together, so we're trying to put together proteins. The process of making proteins here. Proteins are what make up our body. Okay, they make up all the structures in our body. They help with communication in our body, transporting stuff around in our body, in and out of cells. They're enzymes, so they help with chemical reactions in our body. So proteins make up most of what we are. Another name for proteins is polypeptides. So if you see that in a book or in a video or in a worksheet, don't get confused like, whoa, I've never seen this before. It means protein. Proteins are made up of hundreds of amino acids. So we learned that already, that proteins are made up of amino acids. Protein might be made up of 500, 1,000, 1,500 amino acids, depends on the protein. Okay, and there are 20 different amino acids. So if you think about that, there's 20 different amino acids, and a protein is made up of hundreds of amino acids. There is pretty much an infinite number of combinations that you can have for a protein, and therefore you can have all of these different proteins. All right, but let's talk about making the proteins. How do we make it now? Okay, and so the step one here is going to be called transcription. Now you've seen the word transcribe before, but now this is a variation of that word, transcription. But they mean the same thing. Transcribe means I'm going to copy something. Transcribe it, I'm going to copy it down. And so transcription means the same thing. They're going to have to copy something, okay, in this process. And we mentioned earlier that in the central dogma, DNA contains the code for making all our proteins. And we got to make the proteins in the ribosome. And so we got to be able to take that that recipe for how to make the proteins from the nucleus. We got to copy it and take it to a ribosome. And that's what we do in step one here, transcription. And so this is the job mainly of messenger RNA, because messenger RNA have the ability to, they're going to copy the code, and they're like a messenger. They carry that message to where we need it. And so here's how it works. First off, helicase, which we talked about in DNA replication. Helicase is going to unzip the DNA, but different than replication where we unzip the entire DNA, we're only going to unzip the portion of DNA that we need to copy. So the section of DNA that has that gene for the protein we're making. So it just unzips that little part, just the part that we need. And so it unzips the section of the DNA that we need. Secondly, we have something called RNA polymerase. Where in replication we had DNA polymerase, this is RNA polymerase because we are going to be making messenger RNA. And so this is RNA polymerase. And it says here, helps bond the correct mRNA nucleotides to the unzipped DNA, thus making the mRNA strand. So the DNA is going to unzip, and polymerase is going to go and add the mRNA nucleotides to the unzipped DNA. And so in DNA, if I had the nitrogen base A normally would bond to T. But there is no T in RNA, and so the polymerase would put the nucleotide with the nitrogen base uracil, U, with that, because that's their complement. And so polymerase goes and puts all the complementary RNA nucleotides with the unzipped DNA. Now, once we have all those RNA nucleotides bonded to the unzipped DNA, that's what we did. We just copied the code. Then the sugar and phosphate groups on the mRNA have to bond together with ligase. And once that's done, then the mRNA strand is one strand of its own. And so it's going to break away from the DNA at that point, break away, and DNA is found in the nucleus, so it's got to travel from the nucleus to where proteins are made, the ribosome. So that RNA strand will break away, he'll travel out of a nuclear pore in the nucleus, and he's going to go to a ribosome in the cytoplasm, which is called a free ribosome. And if it goes to a ribosome in the cytoplasm, then the proteins that are made are going to be used for stuff within the cell. Now, if we're trying to make proteins that need to go outside the cell, that we need to export outside of the cell, then this mRNA strand is going to go to a ribosome on the rough ER. And the rough ER will send the ribosomes on the rough ER will make this protein. It's going to go through the ER to the Golgi, and the Golgi we know exports things out of the cell for us. So those are the two types of ribosomes that it might travel to. Then we have step 2 here. Step 2 is called translation. So we're translating or deciphering this mRNA code. At this point, we have the mRNA code. The ribosome has grabbed onto it, and the ribosome is kind of like a conveyor belt. He pulls the mRNA code through so we can put the amino acids on one after another, and it also helps to bond all the amino acids together as we put them on there. And so at this point, the mRNA and the ribosome is grabbed onto the mRNA. And tRNA are going to help us out here. tRNA stands for transfer RNA, and they're going to transfer amino acids. And they're going to carry amino acids and attach them in the correct order. And how do we do that? That's where the translation word comes in. It translates it because the tRNA has a 3-letter code on it. We call the anticodon. And the mRNA has 3-letter codes that we call the codon. And so the tRNA carrying the amino acids looking for that 3-letter code on the messenger RNA that match up with his 3-letter code. And that tells him where to put his amino acids. So we can put the amino acids in the correct order based on those 3-letter codes. So as we're putting these amino acids together, the ribosome's pulling the mRNA strand through and bonding the amino acids together. And then once we have all amino acids assembled and bonded together, that protein's going to be done. It's going to break away and travel to wherever it needs to be used. But enough with just the words. It's kind of hard to kind of see what's happening here without drawing it out. So right now we're going to draw out an example of this so you can kind of see how it works. Okay, so here we have our DNA strand. And I kind of skipped ahead here a little bit. Helicase has already unzipped the section of DNA that we need to code for. So this gene, this would be a gene for making a protein. So helicase has unzipped this. We have obviously 2 sides that can get copied, the bottom half or the top half. Well, one of the sides is going to be called the template strand. And so that's the side that we're going to copy, is the template strand. And so this is going to be our template strand here. We're not going to use this strand over here. Now, out here in the nucleus, we have nucleotides floating around. We got DNA nucleotides, we got RNA nucleotides, but in this case, RNA polymerase is going to take the RNA nucleotides and bond them to their complementary letters. So we're going to do this together here. So imagine we have this nucleotide floating around out here. Let's say it was a U, like that. RNA polymerase is going to go and take that U and put it with an A. So it might be right there, or it might be with this A, or with this A. But he's going to go and bond those complements. So let's just say he takes this one over here. And so this U bonds with A. And so I'm going to go through and put all the complementary mRNA nucleotides with the DNA real quick so you can see what that looks like. So we're going to have a U, G, C, C, A, G, C, C, A, G, U, A, U, G. And so you can see here, RNA polymerase bonded all of those mRNA nucleotides to the DNA strand. Next, ligase is going to come in and bond the sugar phosphate groups together. So now, after ligase comes in here, covalently bonds all those sugar phosphate groups together, like that. Now, we have one complete solid strand of mRNA. So the next thing that's going to happen is that mRNA strand, this is mRNA that we made, okay, messenger RNA, it has copied the message. And now it has to play the role of a messenger. So this mRNA strand is going to break away. It's going to go outside of one of those nuclear pores and out here into the cytoplasm. And it's going to find a ribosome. And so let's see what happens next over here. This second step is called translation. And so we have a ribosome here. Ribosomes have two parts. They have a large and a small subunit. And so we have this ribosome, looks like that, and it's going to pull this mRNA strand through it. And so let's go ahead and say we have this mRNA strand, it's getting pulled through there. Okay, it kind of sandwiches it like a hamburger. Okay, and let's go ahead and copy this mRNA code down over here, and so you guys can see what's going to happen here. Let's say this is A, U, G, C, C, A, C, C, A, A, G, U, A, G, U, U, A, U, A, U, G, and those first ones, U, G, C, C. And so here's our mRNA strand, all complete now. Okay, there it is, mRNA. The ribosome here has sandwiched it. It's got a hold of it. This is our ribosome. And let's see what happens next. Okay, so here is what a transfer RNA looks like. Notice it's still single-stranded. Remember we said all RNA is single-stranded. It's one strand, but it folds up in an unusual way. Okay, it folds up in an unusual way so that we have a three-letter section right down here at the bottom. You can see we have this three-letter section right down here at the bottom. That three-letter section right down there at the bottom, that is called our anticodon. Okay? That's what that three-letter section is. That's our anticodon. Now, there's going to be three-letter codes on here that are going to be called our codons. But we haven't determined what those codons are yet, what the three-letter sequences are. I'm going to show you how that works. tRNA is carrying an amino acid with it. Now this amino acid up here, we're going to abbreviate all amino acids. We just use the first three letters to abbreviate them. So this is methionine, but I'm just going to abbreviate it MET. And so even a lot of the amino acid charts that you have, like this, they abbreviate it for you as well. Okay, so that's methionine, MET. There's only one, on this whole chart, there's only one methionine amino acid. It's also known as the start amino acid. Okay, and so there's only going to be one spot for this, and this is always the very first one that we put down. This determines the start of the protein. Okay, and so when this DNA is unzipped, it's not accurate enough to be able to unzip right to the very first letter, right to where methionine starts. It's not that accurate. So when it unzips, there's going to be extra letters at the beginning and end. And so we need to know where to start. And so the ribosome is going to pull this messenger RNA through until it finds a three-letter code that will match up with methionine. And so if you think about this, methionine's anticodon is UAC. And then you have to think about what three letters would match up with UAC. So what would be the complement to U? It would be A. What would be the complement to A? That would be U. What would be the complement to C? That would be G. So we're looking for a three-letter code that's AUG, because that would match up with that three-letter anticodon. And so the ribosome is pulling this through until it finds that three-letter code right here. Okay? AUG. And at that point, this transfer RNA is going to come down and he's going to put his amino acid right there. And so that first amino acid is, here's our amino acid, methionine, M-E-T. And then the mRNA code is going to get pulled through a little bit more, okay? And so that we can find what the next three-letter code is, and the transfer RNA that matches up with that code can put the correct amino acid down. And so that next three-letter code is going to be CCA, and then AGU, and then UAA. Now, we're just doing a small little sequence of mRNA. If this was a real protein, this would be thousands of letters, okay? Maybe not thousands, but 1,000, a few thousand letters. So we're just going to do a small section of this, okay? And this first three-letter code here, AUG, is important also because that determines, from that point on, every 3 letters is going to be a codon. So that determines what our 3-letter sequences are going to be. We call that the reading frame, okay? And so then a different transfer RNA is coming down. He's looking for this 3-letter codon because remember these codes that are on the mRNA are called codons. And so this is where we're going to need the help of this chart. This chart, it can be found in your textbook. You can find these online. You can just do a Google search for amino acid chart. Okay. These charts are going to help you determine what amino acid will go with each of these codons. So for AUG, it was methionine. What would be the amino acid for CCA? So let's go through this together. Hopefully you can see this. I tried to make it a little bit bigger. So our 3-letter codon is CCA. And on this chart, the first letters, so the first letter, which was C right here, corresponds to this left side. The second letter, C, corresponds to this upper part. And the third letter, which was A, goes over here. And so our first letter was C. And so I look on here, my first letter is C. That means it's somewhere in that row right there. The second letter was also C. So I go up here, second letter, C. So that means it's also in that column. So we know it's in this row and this column. So now we've really taken it down. We know it's somewhere in that box right there. And so the third letter was A. So I come across here, A, it's right there. So my CCA is right there. Okay, CCA. And it says here the amino acid is PRO, that is prolines. But like I said, all we're going to do is abbreviate those amino acids. And so my second amino acid would have been proline. And let's do one more here together, okay? This one, the mRNA codon is AGU. So I'm going to look on here for AGU. My first letter is A, so I know it's in this row. My second letter is G, so it's in this column. So now we've narrowed it down to this little box right here, AG. And then U is right here, so AGU is right there. Okay, and that says SER, which stands for serine. And so my next amino acid that transfer RNA would have carried down and put down and helped us place was serine, S-E-R. Now, this next one's really important. Okay, this next one's really important. It says UAA. And so if I look at UAA on here, my first letter was U, my second letter is A. So U and A. So I know it's in this box right there. My third letter was UAA. My third letter is A. So I come across to there, UAA. Now it says stop. Now stop is not an amino acid. This is actually a stop signal. It tells us that this is the end of the protein. Because like I said, DNA will, when it unzips during transcription, it doesn't unzip perfectly. There can be some extra letters. And so that we have to know when is the end of this protein. When do we stop putting amino acids down? And so when you hit that stop signal that says stop, that's the end of our protein. it starts with the methionine, or start one, and it always ends with the stop signal. So in this case here, our protein is only 3 amino acids long. We're just practicing here. It normally would be 500, maybe 1,000 amino acids long. But at this point, these guys would be bonding together. So these guys are getting pulled through the ribosome. The ribosome helps to bond them together, and then this protein would go to where it's needed. So I hope that helped, and we'll talk to you later.